Okay, good morning, everyone. Thank you for getting up this morning to be here for this. We're really excited about the lineup of speakers we have this morning. So I think without further ado, we can get started on the session. And I'd like to invite Dean Islam up to introduce our first speaker. So, Dean.
Good morning, everyone. Thank you, Shay. So it is my absolute pleasure to introduce Dr. Justin Bailey for this session's first speaker. Dr. Bailey received his MD and PhD degrees from the Johns Hopkins University School of Medicine through the Medical Scientist Training Program and completed medical residency at the Massachusetts General Hospital. He subsequently completed fellowship training in infectious diseases at the Johns Hopkins University School of Medicine before transitioning to a faculty position. His primary clinical interests are management and treatment of HIV and viral hepatitis, including HCV. His research interests include the study of human B cells and neutralizing antibody responses against HCV, HBV, and SARS-CoV-2, and respiratory syncytial virus, with a goal of developing improved vaccines against these viruses. So please welcome Dr. Bailey to the stage.
Okay, good morning. Thank you all for coming, since it feels early, at least. And also thanks the organizers and Jordan especially for the invitation to give this talk. This is something that I'm really passionate about, and I know a lot of you in the room are really passionate about this topic, which is why you're here. So this is things that you already know, but there's a new HCV infection in the world every 30 seconds. And that amounts to about 1 million new infections per year, and that's despite the availability of effective treatment. I myself have been treating people for Hep C for the last 10 years. Despite that and everyone else doing the same, we still have increasing incidence of Hep C over time, rather than a decline that we would like to see. So why is there no vaccine for hepatitis C? I think a big reason is what I consider a myth, which is that the genetic diversity of hepatitis C makes a vaccine impossible, or that it makes it even extremely difficult. It's true that Hep C is very genetically diverse, no question about it. These are phylogenetic trees on the same scale of HCV and SARS-CoV-2, which is a respiratory virus that is considered to be genetically diverse. And in fact, to even see SARS-CoV-2 on this slide, the scale had to be blown up 70-fold. So the genetic diversity of Hep C really is enormous. But despite that, there is natural immunity against hepatitis C. About 25% of people who get infected with hepatitis C spontaneously clear the infection without treatment. So, this is the light at the end of the tunnel, right? And even more so, more important than that, what this viral load graph illustrates is that there are some people who clear one Hep C infection and get reinfected, and they're very likely to clear those subsequent reinfections, about 80% likely to clear subsequent reinfections. And in the BBASH cohort in Baltimore, we have individuals like this person shown here on this viral load graph who've cleared multiple infections from multiple different genotypes and subtypes. And work that Bill Osborne and Andrea Cox did, shown in the graphs on the right, show that with each subsequent infection, both the peak viremia level and the duration of infection decreases. So it's really an illustration of protective adaptive immunity in this subset of people. So really, this is the model that we're trying to follow as we think about how we might develop a vaccine that's protective for all people against hepatitis C. So the thing I'm going to focus on today is the fact that we've seen that natural immunity is associated with developing broadly neutralizing antibodies against the virus. What we've seen over years, and the more we look, the more epitopes we find, is that these bNAbs actually target epitopes across E2 and E1. The structures that I'm showing here on this slide on the left show 3 different sites on E2 that are targeted by bNAbs. The front layer on the bottom is a really commonly targeted epitope. We've also found really good bNAbs that target the back layer of E2. And then the structure to the right is our newest antibody that we've isolated. This is actually a computational structure, not an actual structure. But we're pretty confident that this antibody, which is broadly neutralizing, targets a quaternary epitope that actually spans E2 and E1. So again, I think one point that I'll make is there's not one single neutralizing epitope that I would characterize as most important. But the good news, I think, is that there are multiple targets of vulnerability on the E1, E2 protein. Oh, and sorry, one other point that I want to make is especially the front layer antibodies almost always use this particular V gene segment, VH1-69. This is a V gene that has an ortholog in non-human primates, but does not have an ortholog in rodents. And I'll come back to that point. So this is a slide from my friend Ellie that illustrates the vaccines for Hep C that have been tested in humans to date. So I almost changed the title of this slide to "How Not to Make a Hep C Vaccine," right? So in the course of human history, we've tested 2 vaccines in humans. One of those was Michael Houghton's E1E2 protein vaccine that was tested long ago. And the other more recently was a T-cell only MVA vaccine that Andrea Cox and others tested in a phase 2. And unfortunately, that vaccine was not protective, although it did reduce peak viremia in the people who got infected. On the other hand, this is sort of a rough outline of ongoing preclinical studies that I know about and that Ellie knows about. As you can see, there are lots of vaccines and vaccine candidates that are being tested in mice or have been tested in mice or other rodents. A smaller subset that we know of that have been tested in non-human primates. That includes, in particular, work done by Rama Amara at Emory, testing DNA, MVA, mRNA vaccines in monkeys. And then also an mRNA vaccine that Ellie, Andrea, and I are testing currently in non-human primates. And the rest of my time today, I'm going to talk a little bit about the antibody data that we have from that ongoing trial. So first I want to talk a little bit about where our vaccine antigen came from, how we chose our E1, E2 vaccine antigen. And this came from work from Nicol when she was in my lab. And what we noticed was that in people who got repeatedly infected with hepatitis C, some of them developed very broadly neutralizing antibody responses and others did not. And so we wanted to look at what's different about the viruses that these people were getting exposed to that in some cases drove development of bNAbs and in other cases did not drive development of bNAbs. So we characterized the antigenic characteristics of these envelope proteins, these E1, E2 proteins. The way we did that was we measured binding potency essentially of a panel of antibodies, broadly neutralizing antibodies and also germline ancestors of broadly neutralizing antibodies. And those are listed on the x-axis across the top of the graph. And the y-axis is all of the different E1, E2 proteins that we tested binding. when we looked at these patterns of binding, we saw that even though these were really genetically diverse E1E2 proteins, they actually fell just into 4 clades based upon their pattern of antibody sensitivity. And so we call these antigenic clades 1 through 4. And then we look to see, does the antigenic clade that somebody gets infected with influence whether they develop broadly neutralizing antibodies or not? One point I'll make is that when we tried to map these antigenic clades onto the genetic diversity of the viruses, onto the genotypes of the viruses, there was no relationship. So there were genotype 1, genotype 3, 1As, 1Bs that fell in antigenic clade 1 or that fell in antigenic clade 4. So genetic diversity had nothing, or genotype, subtype, had nothing to do with this antigenic diversity. But what we saw was that in the people who developed BNAbs, they were repeatedly exposed to viruses that fell in antigenic clade 1. And that's the boxed graph on the left. So you can see as you go increasing number of infections with these antigenic clade 1 viruses, steadily the neutralizing breadth of the plasma in these people increased. In contrast, if you look, for example, at the bottom right graph, people who were repeatedly exposed to antigenic clade 4, their neutralizing breadth did not increase with these subsequent exposures. And when we developed a model where we incorporated multiple variables that might be associated with increasing neutralizing breadth, the strongest factor was this repeated exposure to antigenic clade 1 viruses. So we chose one of these antigenic clade 1 viruses as our E1E2 vaccine antigen. And that virus is called C110 day 0, or I guess on this slide it's called S110 day 0. And then as a control, we also made a vaccine based upon a clade 4 antigenic variant. The other thing that this slide is showing is that the initial mouse studies that were done with this vaccine were a version of the vaccine that expressed core E1E2 and P7 with the goal of forming virus-like particles after mRNA vaccination. And this is work that was done in Ellie Barnes's lab. In particular, it was done by Gerardo Montalvo in Ellie's lab. So the graph on the left is showing antibody titers against both homologous and heterologous antigens after the mice were vaccinated either with the antigenic clade 1 vaccine or with the antigenic clade 4 vaccine. So antigenic clade 1 titers were higher both against homologous and heterologous variants compared to the mice that were vaccinated with the antigenic clade 4 viruses. So this supported our hypothesis that there's something special about these antigenic clade 1 E1E2 proteins that makes them a good vaccine antigen. So we wanted to carry that vaccine forward, and I want to pause here and talk about the importance of the animal model that we're using to assess vaccine response. I mentioned before that we know that a lot of broadly neutralizing monoclonal antibodies in humans use particular V gene segments, and even in some cases particular D gene segments, and that mice in general do not have orthologs for those V gene segments. In contrast, rhesus macaques do have orthologs, do, for example, have an ortholog for VH1-69. And Manson Law did a really important study, and the graphical abstract is shown here, where he took Michael Houghton's E1E2 vaccine that had been previously tested in humans. He took that exact same vaccine, tested it in rhesus macaques and in mice. And he compared the antibody responses that he got in the mice to the macaques to serum that was left over from the vaccinated humans. What he saw was that the rhesus macaque responses, the antibody responses, much more closely resembled the human responses than what he saw in mice. And he even went so far as to isolate monoclonal antibodies from the vaccinated monkeys and show that the monoclonal antibodies he isolated were also similar to broadly neutralizing antibodies from humans. So I think that this is strong evidence that macaques are a much better model to understand vaccine antibody responses than rodents. So with that in mind, we tested our E1E2 vaccines in non-human primates, in rhesus macaques. And I'll tell you a little bit more about the specific vaccines that we're testing. So I mentioned the core E1E2 P7 mRNA. We're actually mixing that with a genotype 1 to 6 antigen that Ellie developed that's designed to induce T-cell responses. And then we also made a vaccine that expresses the C110-0 E1E2 only, not the core and the P7. And that also was mixed with the genotype 1 to 6 mRNA. So, the goal of this vaccine is to induce both antibody responses and T-cell responses. But I'm just going to talk about the antibody responses today. We also tested each of these vaccines at both a 100 microgram per monkey dose and a 10 microgram dose. So I'll refer to those as high and low doses from here on. This is the vaccination schedule and blood draws that we did in these monkeys. There were 4 macaques in each group, so a total of 16 monkeys between the 2 vaccines and the 2 different doses of each vaccine. We did a prime and 2 boosts with blood draws after each. And I just wanted to mention that all of the antibody work was done here by Jessica Mims in my lab. So here's another thing that I want to talk about is how do we assess the antibody responses that we get in these monkeys after vaccination? How do we know whether we're inducing a good antibody response or a bad antibody response? And so we put a lot of thought into this. And one of the things that we did was we measured neutralizing breadth using a diverse panel of pseudoparticles of a whole bunch of different participants in the BBASH cohort. And that included people who spontaneously cleared Hep C infection and also people with chronic infection. So the people who cleared are in the box at the top, and the people with chronic infection are at the bottom. They're ranked from greatest to least neutralizing breadth of all the people we studied. And the top 10% of people with the best neutralizing antibody responses who cleared Hep C infection we're calling elite controllers. And so we chose 2 of these elite controllers as our benchmarks to compare the macaque results to the antibody responses in the elite controllers. And from among the chronically infected people, we chose the person who fell right at the median in terms of neutralizing breadth as sort of our lower limit benchmark control. So we said we definitely want to get better antibody responses than this median chronically infected person. And in the best case scenario, we'd like to get antibody responses that resemble the elite controllers who spontaneously cleared Hep C. To say a little bit more about the neutralizing panel, I think we'll talk a lot about this in the afternoon, but this was work of a lot of people. Some of the people in this room contributed to develop an antigenically diverse and representative set of HCV pseudoparticles that could be used generally to measure neutralizing breadth and neutralizing potency of either plasma or monoclonal antibodies. And I've talked a lot about the difference between genetic diversity and antigenic diversity. I just want to point out that this is not a set of viruses selected just to represent multiple subtypes or genotypes, which does not dictate neutralizing antibody sensitivity. Instead, we chose variants that have a range of neutralizing resistance, all the way from very sensitive up to extremely resistant, tier 1 through tier 4. And so, the tier 4 viruses are a very high bar in terms of neutralization, and the tier 1s are relatively easy to neutralize. So first I'll show you some results that we got in terms of binding results with our mRNA vaccines. And so always I'll show in terms of the groups of monkeys The low doses are on the left of the graphs, and then the higher doses are to the right. And then the benchmarks, those plasma samples from the elite controllers and the chronically infected people, are on the far right of each graph. And so, again, these are binding results. The top row is homologous E1E2, so the vaccine antigen binding. And the lower row is heterologous binding. So after prime, you can see there's very little antibody response. After boost, the antibody responses start to pick up, in particular in the higher dose vaccine groups. And then after the second boost, we start to see fairly robust binding antibody responses. In particular, against the homologous E1E2, we see antibody titers that are pretty similar to the elite controller benchmarks. If you look at the bottom row after boost 2, we do see okay binding antibody titers after boost 2 against the heterologous E1E2, but they're relatively similar to the chronic median, the lower benchmark control, and not as high as the elite controllers. Here I'm showing neutralizing potency against a heterologous variant. This is actually a tier 1 genotype 5 heterologous variant. So again, after prime, we don't really see neutralizing antibodies in any of the monkeys. After boost 1, we start to see some neutralizing potency. The y-axis here is IC50 reciprocal dilution. But then after boost 2, we see a very nice jump in neutralizing potency, especially in the high dose groups. I'll note that by boost 2, we had dropped out the low dose E1E2 vaccine group because they had shown very poor responses in the earlier time points. But you can see that despite the sort of unimpressive binding titers that we saw in the high dose groups, their neutralizing titers are actually clearly better than the chronic median control, and sort of starting to approach what we saw in the elite controllers. Now this is the really exciting part. Once we measured neutralizing breadth, which here on the y-axis, it's the number of pseudoparticles neutralized out of that panel of viruses that I mentioned. If you look after boost 1, again, you can see that the two high-dose groups have neutralizing breadth that's similar to the chronic median after the first boost. But after boost 2, the neutralizing breadth that we see in the monkeys is clearly better than the chronic median benchmark. And in a couple of the monkeys, in the high-dose E1E2 group, we see even better neutralizing breadth in the monkeys than we saw in the elite controller benchmark plasma. So overall, after boost 2, the neutralizing breadth in the two high-dose macaque groups is pretty similar to the neutralizing breadth that we saw in the elite controller benchmarks. This is another way to look at those same data. Here now each dot is an individual pseudoparticle, and the pseudoparticles are colored by whether they're tier 1 to tier 4. Tier 1 pseudoparticles are red, and tier 4 are blue. The dashed line on each graph is the threshold of what we consider positive neutralization. And now we've got the monkeys broken out by individual monkey within the vaccine groups. So the thing I want to point out is that if you look at the high-dose E1E2 group on the far left, there are some monkeys who neutralize almost every pseudoparticle in the panel, and that includes tier 3 and tier 4 pseudoparticles. And you can look on the far right to see the same assay done in parallel with the plasma from the elite controller benchmarks. And you can see that the overall neutralization we're getting in some of these high-dose macaques is similar to what we saw in the elite controllers. And then this is finally one more way to look at those same data. Again, each dot is a pseudoparticle. And now we're looking at the correlation at the level of each pseudoparticle and how much neutralization we saw in the monkeys compared to how much neutralization we saw with either the elite controller benchmark plasma or the chronic median benchmark plasma. So the top left graph is the high-dose CORI-182P7 vaccine compared to the elite controller plasma. And so you can see there's a surprisingly strong correlation between the results that we got at the level of individual pseudoparticles. The same thing was true with the high-dose E1E2 vaccine, where you see a really strong correlation between the macaque results and the elite controller neutralization results. And then the bottom row is the same comparison between the monkeys, but now with the chronic median plasma benchmark. And there's still a correlation, and I think a lot of that is just driven by the tier 1 through 4 pseudoparticles. But the correlation is much worse than what we saw with the elite controller plasma. So overall, qualitatively, the antibody responses in the macaques and the high-dose groups very closely resemble the antibody responses that we see in the elite controller benchmarks. So to conclude, I just want to emphasize that HCV E1, E2 antigenic diversity is much lower than the genetic diversity of the virus. And for antibody response, I think antigenic diversity is what we care about, not genetic diversity. On the T cells, completely different story. But for antibodies, I think it's antigenic diversity that matters. Sorry, I have a lag here. Okay. There are many promising B-cell vaccines in the preclinical pipeline. But we really need more non-human primate studies and more human studies to really know whether these vaccines are likely to be protective. And again, you know, there's that beautiful paper from Manson Law showing that you get more predictive responses from non-human primates. I think the data I just showed you today also show that rhesus macaques can be a very good model to predict, hopefully predict, responses that we would get in humans. I think that we need to always take rodent data with a very big grain of salt in terms of, you know, exactly the nature and the characteristics of the antibodies that are being induced, because rodents don't have these important orthologs of human V genes. The vaccine that we're testing, I think I showed today, induces broadly neutralizing antibodies at levels that are similar to our elite controller benchmarks. The profiles, the antibody neutralizing profiles, are highly correlated between the monkeys and the elite controllers. So overall, I would say the quality of the antibody response that we've induced is really good. I mean, we were really, frankly, surprised at the quality of the antibody response that we got with this vaccine. I do think that the quantity of antibody could be improved, that the titers of antibody could be improved. And so I really think that that's something that we need to work on moving forward. So I just want to thank everybody who's worked on this project. This is a picture of the people in my lab. I, of course, want to acknowledge Andrea Cox, who's my partner in all of this, Stuart Ray, other people in our U19 group, Andrew Flack, Ellie Barnes. Moderna made the vaccines for us. And thank you all again for attending.
Is this on? Can you guys hear me? Okay, perfect. Thank you for that presentation. So I'll ask you to hold questions until after the next speaker, and then we'll have sort of a panel Q&A at that point. So Dean, if you could introduce our next speaker.
Hello again. I'm sure you all are enjoying this session as much as I am. I'm going to introduce our next speaker, Dr. Naglaa Shoukry. Dr. Naglaa Shoukry obtained her pharmacy degree from Cairo University and PhD in immunology from Nicol University. Her postdoctoral research has established the essential and complementary roles of CD4 and CD8 lymphocytes in resolution and protection from HCV. Since joining the University of Montreal and its affiliated Hospital Research Center, CRCHUM in 2005, she has established a translational research program focused on studying immunity to HCV, immunological mechanisms of liver fibrosis progression, and liver cancer. She has been awarded a lot of essentially awards for her distinguished work. So I'd like to mention a couple which I think it's important. She's been selected as a Professor of the Year by the Department of Medicine, University of Montreal, and was awarded 50th Anniversary Recognition Medal by Canadian Liver Foundation. And she has also been awarded an inaugural Rosalind Franklin Award in Science in 2021, and also been recognized for her outstanding contribution from, this was an award for outstanding contribution from women and minorities. And she also been awarded a Distinguished Service Award in 2023 from Canadian Association for the Study of the Liver. But most excitingly, which it is very exciting to me to know that she also be a director of the Canadian Network of Hepatitis C, CanHepC, from 2015 to 2023. So a fun fact about Dr. Shoukry is that she loves baklava and she loves to cook baklava. So whoever has sweet tooth, please contact her. So please welcome Dr. Shoukry. Thank you.
Thank you, Dean. So just to follow up on that with Anna, we were saying that, we can use that for fundraising for CanHepC. I did it for our institute fundraising. We calculated the time spent writing grants versus making baklava. It's more cost effective to make baklava. Okay, well, thank you. Okay, so I guess I see it here. Okay, well, it's great to give this talk after Justin because he already mentioned a lot of the background, so you'll find a lot of overlap, but I'll try to skip quickly over that. So first I want to start by the land acknowledgement. I work in Montreal, Tihoetaki, and I'm sorry if I'm not pronouncing the names correctly, but I try to do my best, which is a place that has long served as a site of meeting and exchange among many First Nations, and I recognize and respect them as the traditional custodians of the land and waters on which I work today. So I think this group doesn't really require a lot of introduction about immunity, but also I try to use reviews as references because I don't want to miss anybody's excellent work, but definitely many of the people in the room has contributed to this knowledge. So one of the main players in immunity to viral infections in general, I don't know if there's a pointer here. Oops, okay. Okay, let's just stick to that. Are cytotoxic CD8 T cells that are able to kill hepatocytes that are infected with the virus, but they cannot do that on their own. They require help from CD4 T cells that provide Th1 type cytokines and other that enhance their killing capacity. I think the experiments that we did with Chris and Arash, where we did CD4 and CD8 depletion in chimpanzees, this clearly showed the importance of these two cell subsets for elimination of hepatitis C infection. CD4 T cells also produce other cytokines like IL-21, especially by a subset known as TFH, that helps B cells to produce neutralizing antibodies that Justin just gave a very nice talk about, that are important for neutralizing viral particles. I would also like to mention that IL-21 also acts on CD8 T cells and enhances their capacity, their cytotoxic capacity and polyfunctionality. And there was an elegant paper from Chris's lab recently in JCI demonstrating the expansion of this subset in chimpanzees, in blood and liver, coincident with increase in ALT and right before viral clearance. So all of these factors work together to mediate spontaneous resolution, and the cells form long-lived memory cells. Antibodies, however, in people who clear, although they do appear quickly, but most of the time they are transient, they are more short-lived than T cells. This does not mean that they are not important, it's just that they have different kinetics in those who clear. In contrast, people who go chronic, which is approximately 70% from those during acute infection, we see dysfunction in CD4 T cells, and this leads to compromising their helping capacity for both CD8 T cells and antibodies. So CD8 T cells express an exhausted phenotype with increased inhibitory receptors, lose effector function, while B cells, they become activated, they express an atypical phenotype, and although the broadly neutralizing antibodies are delayed in those who become chronic, as people become more and more chronic, you start to see better neutralizing antibodies. So it's a question of having the right antibody at the right time. Now we have DAAs that can cure almost at 100%. However, when we treat people who are chronically infected, this does not completely restore the immune response. So there's only partial restoration of the TFH part of the CD4 T cells. Also for CD8 T cells, they express a memory-like phenotype with reduced cytokine production, and they have an epigenetic imprint, what we call the epigenetic scar, that does not allow them to fully recover. On the B cell side, there's reduced activation, but HCV-specific memory B cells persist. And there's decline of neutralizing activity. So it is important to consider if in these individuals will restored memory responses be functional upon vaccination. So evidence of protective immune memory. So Justin has already presented that, so great minds think alike. But it's a great study from the BBASH cohort showing that upon re-exposure and reinfection, most individuals who have resolved one infection will resolve with faster kinetics. So the duration of viremia is reduced, as well as the maximum HCV RNA, and the rate of clearance at this condition becomes more around 50 to 80% rather than 25% during primary infection. So can reinfection studies inform vaccine development? So this is an approach that we took over the past I would say 10 years, where we believe that the continued risk of reinfection in these high-risk individuals represents what we call a natural re-challenge experiment. It can inform correlates of protection in real-life settings, and it can provide benchmarks for evaluation of efficacy of novel vaccines. And this is work that we were able to do thankfully to the great collaboration we've had with Julie Bruneau over the past 21 years already. So it has been very productive. Nicol Flores from my lab will be giving a talk later. I think you are the next talk, about recent work that she's doing with a single-cell RNA-seq to try and identify signatures of protective immunity. So I'm not going to steal her thunder. I just wanted to show that this was the first paper we did on reinfection, and it is a simple graph here. What you see on top, the SR are subjects who have cleared twice, and this is showing the expansion of HCV-specific T cells upon reinfection, which you can see with the dashed line. The bottom graphs are those who cleared the first infection but not the second infection. We call them SRCI. And you can clearly see just by looking at the magnitude that in the ones who do not resolve the second infection, although you have memory cells, they do expand but not to the same magnitude. We've also done targeted sequencing for epitopes that were targeted in these individuals, and we could show in that paper that the autologous virus of the reinfection was not being recognized. So it's important to, when we talk about diversity and homology, to think about these issues with vaccination. I don't have time to show all the data, but also these cells were highly polyfunctional, meaning that they can produce more than one cytokine or effector function. In another study that we published in 2024, where we looked at both the T cells and the B cells and the neutralizing antibodies, we wanted to see the crosstalk in between the two. And what you see here is just the summary of the entire paper as a heat map. And you don't have to go into details. We looked at antibodies titers by ELISA. We looked at breadth and potency, again, using the panels provided kindly by Justin. We looked at B cells using E2 tetramers provided by Arash, as well as the TFH cells that are important for antibody production and the total T cell response. And the only thing you just need to take home from that is that if you look on the, to me it's the left-hand side, the SR-SR, you see a lot more red in that group versus the SR-CI. So it's not like there's an all or none response. It's just that in the ones who resolve repeatedly, it seems that all the stars are aligned properly at the right time where you have everything working in synergy. When we looked at the correlation between the different factors, what you see here, again, with the SR-SR, there are a lot more correlations between the E2 specific B cells, activated CTFH, neutralization breadth, and potency, versus in the SR-CI. You see very little correlation, and this is if you look early on during acute infection. So what about vaccines? So this is a very nice table. It's from a recent review published by Jake Liang, and I like it a lot because it summarizes everything that was done. So I highly recommend that you go check it out. As you can see, there's only one vaccine who made it into a phase 2 trial, which is the non-structural proteins. It's the VIP trial that was performed by the Johns Hopkins group, and I'm going to talk a little bit more about that. So it is a chimp ad prime MVA boost, and this is a concerted effort with Ocarius, with Ellie Barnes from Oxford, Andrea Cox in Johns Hopkins. And the phase 1 clinical trial was actually very promising, and it showed a very high level of immunogenicity and polyfunctionality in healthy donors. Again, healthy donors, if I remember correctly, they're young Oxford students, so they're perfect. And this is showing the immunogenicity. So again, this is data from Ellie showing that with the boost, you see an increase in the interferon gamma producing cells. And also you can see decent cross-reactivity, maybe a little bit between, because the vaccine is a 1B, there is good cross-reactivity with 1A, a little bit less with 3A and 4A, but there is some cross-reactivity. So this set the stage for the trial that was run by Andrea and Kim Page, who I think will be talking about this in the next session, of PWIDs who are the population that is most at risk of HCV infection at 3 clinical sites. And just to summarize, there were 2 arms, a vaccine arm and a placebo arm, and the endpoint here was not prevention of infection, but prevention of chronicity. However, in both groups, it was exactly the same number, 14 individuals developed persistent HCV. However, there's always hope, because the ones who were vaccinated did have a lot of T cell responses, but also they had lower viral loads, as also mentioned by Justin. So can we understand this response better? So some of the work that we tried to do, and we kindly were provided data from Ellie in order to do that, is that we wanted to see how this compares to a reinfection. So we looked at transcriptomic signatures in resolvers during both primary infection, reinfection, and data that Ellie had from one of the phase 1 trials of the chimp ad vaccination. We reasoned that a primary infection would resemble what you would see with the vaccine prime, while secondary infection would resemble what you would see with a vaccine boost. What we observed is that during primary infection, there's a lot of induction of innate immunity, not so much during secondary infection, and this is just comparing infections, not the vaccine. There's also induction of B cells and B cell memory. And there's also induction of follicular helper T cells and CD8 T cells. When you add the vaccine on top of that, you see that there's this glaring empty space, which relates to B cells that were not part of the vaccine. So that could be one of the possibilities that the vaccine did not induce neutralizing antibodies, but definitely it did induce very strong T cells. So what I want to conclude for that part is that for the next generation vaccines, a combined antibody and T-cell vaccine is probably the best. There are still lots that needs to be done, and I know Andrea is working on that, trying to understand why things did not work with the vaccine, whether it is virus variability, reduced immune responses in PWIDs, or others. So what else is in the pipeline? So I just want to focus on three things in the pipeline. So the first one is a clinical trial that will start soon in Canada, led by Michael Houghton, Sir Michael Houghton. This is scheduled to start in April, and it is the E1, E2 vaccine, but with a new adjuvant. So it will still be targeting antibodies, but what Mike is working on is that he's also trying to generate T cells. So his group have designed a multiple highly conserved CD4 and CD8 T cell epitopes, and these are slides from Mike, as synthetic peptides. And these peptides can be presented by multiple HLA alleles that cover 90% of the population. And although these are epitopes that were designed for humans, you can see here at all the circled populations, when you vaccinate mice, then they produce very nice cytokines, TNF alpha, as well as interferon gamma. So this is something that would be added to that vaccine at a later stage. The second, I think Justin alluded to that as well, is this very nice study that came from Ellie's lab, recently published in Hepatology, where in order to overcome the variability of the virus, they looked at two strategies. The first one, to use conserved segments, genotype 1 to 6, and the second one is to do heterologous vaccination, starting with a genotype 1A followed by a genotype 3A, so a chimp ad prime, followed by an MVA boost. The top strategy produced T cells that are cross-reactive to different HCV genotypes. The bottom strategy with heterologous genotypes induced both cross-reactive T cells specific to the genotype 1A, as well as T cells specific to 3A, which is the second, which is the boost strain. So, but again, this is still in mice, so there's still work to be done. And finally, I wanted to talk about work from Rama, who is part of our U19. So he's the one who led that project. And it's a paper that will be coming out soon in Hepatology, where he used DNA MVA vaccines, and he did vaccinations in both mice and rhesus macaques. For the sake of time, I'm just showing this is the structure of the vaccines, so non-structural proteins as well as structural proteins. And I'm just showing the macaque data where he had different groups that received either one of the two or both. And again, for the sake of time, what I just wanted to show is that within that study, the macaques generated a strong T cell responses, both interferon gamma, CD4, positive CD4 T cells and CD8 T cells against multiple proteins within the vaccine. And when we compared that with HCV resolvers from our HepCo cohort, which you see here in orange, you can see that the response is very comparable, sometimes even higher than what you see with HCV resolvers, and definitely a lot higher than chronic HCV. So other considerations, there are many other alternative platforms, mRNA, viral-like particles. So these are all things that are in the pipeline. Different adjuvants can also be used. We had a discussion about animal models. So the rhesus macaques seem to have, from the T-cell side and the antibody side, a good response, but you still cannot re-challenge them. Now we don't have chimps. There are the rat Hep C viruses where you can challenge. So these are interesting, but you would have to make your vaccines in that construct, but it is still something that could serve as an important preclinical model. So there's still work to be there. And of course, the chimp. And we also have to think about vaccination in DAA-cured individuals and whether vaccination in that group will give the same response. So finally, I just want to thank all members of my lab, all the collaborators. And this is a photo from our annual barbecue. If you're ever here in the summer, that's when you get to eat the baklava and all the nice goodies. Thank you.
Thank you very much, Naglaa, for an excellent presentation. Justin, if you can come back, we'll have you both sit here. Yeah. Okay, so if anybody has any questions, now's your chance. Okay, Adam.
Thank you both. Very informative talks on both of those. I'm going to start off a question for Justin. When you look at the sort of durability of these broadly neutralizing antibodies versus the non-neutralizing antibodies, is there any difference in these reinfection patients that are spontaneously clearing multiple times?
So the interesting thing that we've seen in terms of antibody titers in plasma is that in both the people who clear reinfections and in people who clear just one initial infection, the antibody titers drop pretty rapidly after clearance. In as far as we've looked, we do see steady, strong persistence of E1, E2 specific memory B cells over the long term. We haven't done what I'd call a really careful comparison between, in that regard, with the memory B cells, comparing chronically infected people to people who clear infection to really understand the meaning of the persistence of the memory B cells. But I think, I guess I'll leave it at that. Yeah.
Great talks, both of you. Jason Grebley from Kirby Institute. I was just curious, I have a question for Justin. In terms of the dose for the sort of, do you think that, like, I'm just curious on your perspective of, do you think we can go higher on that dose? What are some of the considerations around that?
The dose of the mRNA vaccine?
Yeah.
Yeah, I mean, 100 micrograms is a pretty big dose.
Yeah.
So probably to translate that to humans, we would need to come down a little bit. And that gets back to my point that I think we need to find a way to more efficiently induce high titers of antibody. So, you know, even with a 100 microgram dose, I would have liked to see higher titers. And so, and you know, at the lower, the 10 microgram dose, there was a dose effect already. So I do think that we need to find better adjuvants potentially, or more stable antigens, or potentially combinations of antigens that might keep that quality of the response that we saw, but boost the titers and maybe also allow us to reduce the dose. Because I think 100 is pretty high.
Yeah, awesome. Thank you.
I was going to ask something similar, not so much about the dose, but about the persistence of the durability of these responses. Because I think one of the big questions for us is, how often will you need to vaccinate someone? And this is particularly, you know, an important question for thinking about the population. So I'd be curious for both of you to just a thought about what you do think will be required in terms of maintaining a very long-lasting response, and adjuvants being an important question to that.
Yeah, so we still plan to look at a 6-month time point in these macaques to see how well the antibodies are maintained, and also we're going to look at B cells to see how well the B cells are maintained. I mean, I think for a vaccine to be practical, we can't be reboosting people that frequently. My hope is because, again, in the reinfection people, titers do drop, and nevertheless, they're protected against reinfection. My hope is that we'll see durable maintenance of memory B cells that will be protective, you know, kind of like the way that your Hep B titers can become undetectable and you're still protected against hepatitis B. But all that remains to be seen.
Do you have a thought in the T cell side?
We know that at least from human studies that T cells can survive up to 20 years, and this is early work from Barbara Rehrman. So I'm a T cell immunologist. I have more confidence in T cells. It's not that I don't like antibodies. I've converted. So, but I think that all the data from the human studies point to the fact that T cells in general are more durable than antibody responses. So I don't think that for T cells in particular, we would have to boost as often.
All right, I'm going to direct this question to Naglaa because it's coming on the T cells. I mean, the transcriptomic data was really interesting that there was no second innate signature upon the reinfection. Is that, do I recall that correctly from the heat map?
Yes, that is correctly. So what we think is that maybe for the reinfection, that everything happens so fast that we may have missed the increase in innate immunity that you see with the primary infection.
In those patients that cleared the first time and went on to chronic infection, do you see different profiles in their innate response after that first or second? I can't recall what it said, or changes, differences in ALT that would reflect different degrees of intrahepatic immune activity.
So ALT, we don't see a lot of differences actually, but you know, we don't look at ALT on the long run, but during acute infection, we don't see a lot of variations in ALT. And I think Nicol will present data about the CD8 T cells and their single cell profile in those who clear versus chronic, so I'll let her answer that.
Thank you.
Hi guys, thank you so much. I know we're going to break out into different groups, but I wanted to, when we're all together, Naglaa, you brought this notion, and I think, Justin, if you can also talk about this, this notion about scarring, so that when an individual is infected, exposed, infected, and let's say for 6 months you have this persistent infection, now with all these CD8 and CD4 responses that are having changes in their sort of, you know, genetics. Thank you. Yeah. As a thing going forward for the CHIM studies, what are your perspectives on that? These individuals are going to be exposed and potentially altering their T and B cell responses, and I know you're going to come in with DAAs and so forth, but how should we be, you know, more concerned about this, that we are changing, altering these things or not?
Take that, or should I? You go.
I think in general, one thing I'll say is that in people who spontaneously clear Hep C, we don't see any long-term adverse outcome, you know, increased risk of HCC or cirrhosis in people who clear acute infection. So I think that's the number one reason that I don't worry about causing harm with CHIM if we treat people within, say, 6 months or a year of infection. I think that these immune scars are interesting. I think it would be fantastic if we could see longer-term follow-up in some of these studies to know how long it takes for these things to return back to normal, because I wonder whether calling it a scar is a little bit pejorative, and really we should just call it a mark of recent infection, and maybe that's something normal that would return to baseline over time.
I would just like to add that these studies were mainly done on people who have been chronically infected for years. Right now, there are a lot of studies that people are working on. There was a very nice poster from Jordan's group with Ayub here, looking at early treatment, so treatment during acute infection. And my prediction, and I don't want to speak for you, but I think by treating early, you will avert a lot of these backgrounds or of these side effects, if we can say that. But we do have to do the study and find out. Ali?
Yes, very related to that. Naglaa has got data on scar, which is not scar.
Yeah.
And I agree, we shouldn't be using this in acute infection, and it isn't there after acute infection. There is no scar.
Yeah, so actually, Georg's data, he looked at epitopes that mutate early on, and in these individuals, the epigenetic changes have not set in yet. So.
Hey, this is Rajiv from NIH. So I used to be a B cell person, but I've also changed. So I guess like we need both arms. So there is no way like we can be exclusive. So just in like with your titers and IC50, IC80, can you make some correlation, like some prediction for viral control? I guess you might have planned some challenge studies or something. So is there any way or like, because I'm thinking ahead, like what can be the benchmark sort of thing, like in the elite controllers or all this stuff? Yeah.
Yeah, I think if you just try to extrapolate the IC50s or IC90s that we see with plasma, even in elite controllers who repeatedly clear Hep C infection, the numbers don't really work. So, you know, even in the elite controllers, we're not seeing this super potent neutralization that you would say, oh, clearly this would just immediately eradicate infection because you have 100% neutralization in vitro. The amount of neutralization that we're seeing, even in the elite controllers, is much lower than that. But I think one of the things that we've seen that's really interesting is that in these elite controllers who clear infection, we see that their antibody response drives sequential evolution of the virus that leads to loss of fitness of the virus over time. And then that ultimately leads to clearance of infection. So we're looking at something a lot more nuanced than just absolute neutralization that's leading to clearance in natural infection. And so I think rather than aiming for a specific IC50 or a specific IC90, I think we really should just be directly comparing head-to-head with the plasma of these elite neutralizers. And I think that if we can achieve that level of neutralization, that that's probably the best we can do, and that hopefully, probably, that would be protective in humans.
We'll take two more quick questions, and then we'll
Yeah, this is Jake Lian from NIH, and I really get a sense of excitement about the prospect vaccine among here. So I think this is great because we all know that vaccines being the wilderness for the last 2 decades in the HCV field. And I don't have to preach to the choir about the importance of vaccines, right? We're all here. The only thing I would encourage the whole field to do is to try to get into the phase 1 trial as soon as you can, because I think that's the key, right? You keep pointing out the difference between mouse and primates and humans. So if we can get into the human, that's where we're going to get the data. And also, I think the whole thing with the chimp is that if we don't have a vaccine to test, then the chimp is really useless, right? So I would encourage the whole field to think about that, how we can get that into the phase 1 trial. As Naglaa pointed out, there's only one in the phase 1 trial. Even the FDA, I've been talking to the FDA, they all said that why is there no application for a phase 1 trial? I think that's really something is really, well, it's money, yeah, but I know there's been a lot of confusion in the U.S. about vaccine these days, right? But that shall pass. So my point is that I think there's a great prospect and we should keep pushing at it. And I certainly applaud all of you for continue to focus on these critical tools and knowledge that we can gain. So that's just a comment. Thank you.
Jens Buch from CanHepC at the University of Copenhagen. As you all know, I'm a very big proponent of the importance of neutralizing antibodies. I do, however, think that, and I would like your comments on, that we don't really know what the nature is of the protective immunity following reinfections. Is it antibodies? Is it T-cells? We certainly know from chimp studies that you can clear repeated reinfections without having even autologous antibodies following challenge. So I would like your comments on that.
I think what we've seen in BBASH is also that there are some individuals who do seem to clear despite pretty poor antibody responses and strong T-cell responses. On the other hand, we have individuals who clear with apparently poor T-cell responses and very strong antibody responses, but the majority have both. So I think that in the interest of trying to make a vaccine that's universally protective, we really should focus on trying to induce both good T-cell responses and good antibody responses.
Did you also have a comment?
Well, I completely agree. There's a graph or a depiction I used to show. I think it's a continuum. So there will be some people who are at one end of the spectrum who have very strong T cells and will clear just with T cells, while there will be others at the other end of the spectrum who have strong antibody responses and clear with just antibodies. But most people will be in the middle. And I think the key question here is the variability of the virus. So if your T cells recognize the virus that you are infected with, they will be very efficient. If not, then they are not as efficient, and you need antibodies to come in and help them, and vice versa.
If we have time, can I make a quick comment?
Very quick.
Andrea Cox from Johns Hopkins. I actually think, and we haven't published these data, but we have data in both the VIP trial and the BBASH cohort that it's not breadth and magnitude, it's a nature of the T cell. So I think there are actually really important characteristics, probably of B cells, but also of T cells that determine outcome. It's not breadth and magnitude, it's much more complex. And we see, and we'll talk more about it this afternoon, but we have more data and it's not so simple. But I agree that we should be aiming for both.
Yeah, and just one quick more question. So the nature of those antibodies, do you think they could be very different when you test right after the acute infection compared to later? Also with respect to type of antibodies, the breadth. I think there's some evidence of that. Do you want to comment on that?
Sorry, you mean?
Yeah, that there might be antibodies in the very early acute phase that just is very different in type and also breadthness compared to later in the infection.
You mean later in chronic infection?
Yeah.
Yeah, I think lots of folks have shown that antibody titers increase over time, and antibody breadth increases over time with chronic infection. And we saw that as well in our model, that time was associated with breadth of neutralization. Notably in our model, it was a weaker factor than this antigenic clade 1 exposure in terms of what was driving breadth, but time is definitely associated. And the other thing that's interesting is that it does seem like the antibody response focuses more and more on this front layer over time, that most of the broadly neutralizing antibodies in chronic infection seem to target this one particular epitope in the front layer of E2. I mean, we still plan to do this in a much more comprehensive way, but what we've seen so far in acute infection in people who clear is that they have a much greater diversity of antibody response targeting epitopes across E1, E2. And in particular, what we're seeing now are a lot of antibodies that are E1 dependent, meaning they only bind when E1 and E2 are co-expressed. So I think that that's still an open question and still really interesting and something that's important to figure out.
Okay, so thank you to both speakers for fantastic talks and excellent discussion. So we're going to move into our selected abstracts now, and we'll continue on the T-cell theme with the presentation from Nicol Flores from Naglaa Shoukry's lab. And Nicol will be presenting on IL-34 expressing CD8 T cells and characterizing protective immunity in HCV infection. Nicol.
Okay. Well, good morning, everyone. Thank you for being here. So, as we know, HCV infection has two different outcomes. Around 25% of people who get infected eliminate the virus on their own, what we call spontaneous resolution, while the other 75% develop chronic infection which can lead to chronic liver disease and even liver cancer. How? Sorry. Okay, it's good, thank you. However, we know that people who spontaneously resolve HCV once are more likely to do so in case of reinfection, which shows that a spontaneous resolution of HCV can provide long-term protective immunity. And this is very important because even though we have highly effective treatment to cure HCV, we don't have a vaccine, which remains essential to achieve the World Health Organization's goal of HCV elimination by 2030. In this context, HCV reinfection serves as a natural model of post-vaccination viral re-exposure, meaning that by studying reinfection, we could identify the correlates of protection that an effective HCV vaccine candidate should elicit to prevent chronic infection. Our team and others have shown that a broad and polyfunctional HCV-specific CD8 T-cell response is essential to spontaneously resolve HCV, while in people who develop chronic infection, HCV-specific CD8 T-cells get exhausted and they lose their effector functions because they're in constant contact with the viral antigens. However, the molecular mechanisms that distinguish functional from dysfunctional responses remain unclear. That's why in this project, our aim was to investigate the long-term CD8 T-cell immune response during HCV reinfection to identify correlates of protection that could guide HCV vaccine development. We hypothesized that the spontaneous resolution of HCV reinfection was dependent on the expansion of a unique set of HCV-specific CD8 T-cell clones that preferentially activates pathways that promote antiviral effector functions and polyfunctionality, and that would represent a core protective signature against HCV. So to do this, we evaluated HCV-specific CD8 T-cells from participants of the Montreal Hepatitis C cohort. Those participants are people who inject drugs, so they are at high risk of getting infected with HCV. And we took blood samples from people who had spontaneously resolved HCV at least once and then got reinfected. And we grouped them based on the outcome of reinfection. So in the first group, we had people who spontaneously cleared the virus again upon reinfection. And in the second group, we had people who developed chronic viremia even though they had cleared the virus in a previous infection. And we took samples at 2 different time points of reinfection: early acute, which is the first 3 months after the estimated date of infection, and follow-up, which is around 1 year after the estimated date of infection. So to identify the HCV-specific CD8 T-cells, we used an activation-induced markers assay, or AIM assay, in which we stimulated PBMCs with HCV peptide pools during 18 hours. And at the end of that stimulation, we sort CD8 T-cells that upregulated activation markers, specifically CD69, 4-1BB, CD40 ligand, and OX40. And those were considered HCV-specific CD8 T-cells. After sorting them, we performed single-cell RNA sequencing and T-cell sequencing to have a high-resolution transcription profile of those cells and be able to compare both groups, resolvers and people who develop chronic viremia. After doing the transcription analysis, we identified 10 clusters of HCV-specific CD8 T-cells and 1 cluster of Mait cells. Mait cells are not virus-specific, but we believed they got activated during the stimulation assay in a TCR-independent way. All of these clusters were annotated based on their expression of canonical CD8 T-cell marker genes, as you can see in the heat map here. And we wanted to see if the distribution of those populations was different across groups. So indeed, we found that in resolvers and people with chronic infection, both at early acute and follow-up, the proportions of those clusters were different. Specifically, at early acute reinfection, HCV-specific stem cell memory and effector memory CD8 T-cells were upregulated in people who resolved reinfection, while HCV-specific early effector CD8 T-cells were upregulated in the chronic group. So early effector cells are a subset of CD8 T-cells that proliferate quickly, but haven't yet differentiated into either short-term effector cells or long-term memory precursors. So we wanted to evaluate the differentiation trajectories in our data set. So we perform an RNA velocity analysis that allow us to identify progenitor-progeny relationships between the cells, between the subsets. And we found 2 main trajectories, one going from early effector to effector memory CD8 T-cells, and another one going from early effector to short-lived, to effector-like exhausted CD8 T cells. Both trajectories were present in resolvers and in the chronic group. However, we hypothesized that the early effector memory trajectory was favored in the resolvers, while the early effector and going to effector-like exhausted trajectory was enriched in chronics. So to answer this question, we looked into the clonal enrichment and connectivity of our cells. So here I'm showing circus plots. Each arc or section of the plot represents one cluster, and the bands connecting two clusters represent the number of unique CD8 cell clones that are shared between clusters. So here we can see that there is an enrichment of effector memory CD8 cell clones in resolvers compared to chronics. And in chronics, there are more effector-like exhausted CD8 cell clones. And this is both through at early acute reinfection and at follow-up. So this supports the idea that differentiation towards memory is favored in resolvers, and that differentiation towards exhaustion is favored in chronics. And this is evident even at the early acute phase of reinfection. And when we look at the overall polyfunctionality and exhaustion signature scores of the cells, we can see that HCV-specific CD8 T cells in resolvers are more polyfunctional and less exhausted than those in people with chronic infection. After this, we wanted to see if there were specific gene sets enriched in one of the groups. And indeed, we found that oxidative phosphorylation was higher in HCV-specific CD8 T cells from chronics. And this is interesting because the regulation of metabolism is related with the emergence of exhaustion on CD8 T cells. And interestingly, when we looked at the oxidative phosphorylation signature of CD8 T cells in the effector-like exhausted cluster, the OXPHOX score was lower in resolvers at early acute, showing that even in this population of cells, HCV-specific CD8 T cells from resolvers were more functional than those from chronics. Next, we wanted to investigate more Mait cells. So we saw that the proportion of Mait cells was higher in resolvers, both at early acute and follow-up. And these are a subset of CD8 T cells that have an invariant TCR. They usually recognize bacterial metabolites, but they can also get activated in a TCR-independent manner through IL-18 and IL-12. So that's why Mait cells are activated during human viral infections. And importantly, in chronic HCV, Mait cells are diminished both in blood and the liver. So first, we measured the levels of IL-18 in the supernatant of the AIM assays, and we found a positive correlation between that and Mait proportion. So that means that Mait cells were activated through IL-18 during the AIM assay. And after that, we looked at the expression of different Mait and cytotoxic genes in Mait cells. And we found that Mait cells of people who resolved reinfection at the early acute phase were more cytotoxic than those from the chronic group. So this suggests that Mait cells might be contributing directly to viral clearance. Finally, we looked at the differentially expressed genes between both groups, resolvers and chronics. And at early acute, we found many differentially expressed genes. We then found many at follow-up. And first, what we can see is that there were genes related to cytotoxicity, like granzymes and granulosime, upregulated in resolvers at early acute. But interestingly, we also found a constant upregulation of the cytokine IL-32 in resolvers. So here I'm only showing 3 clusters, but this upregulation was shown in most of them. So IL-32 is a cytokine that is related to inflammation, but its role in the spontaneous clearance of HCV is unknown. So we wanted to investigate this further. And first, we look at the expression of IL-32 in each one of the HCV-specific CD8 cell clusters. And we found that short-lived effector cells, effector memory, and stem cell memory were the populations with the highest expression of IL-32, while the effector-like exhausted subset had the lowest expression of IL-32. So this might mean that IL-32 is associated with the effector function of CD8 T cells. And confirming our previous results, HCV-specific CD8 T cells had an overall higher expression of IL-32 compared to cells from the chronic group. And since this cytokine has 9 different isoforms, we measured 6 of them by RT-qPCR, and we found that IL-32 beta was the most predominant isoform, both in resolvers and chronic. So this opens a new pathway of investigation related to the spontaneous resolution of HCV. In conclusion, we found that people who resolve reinfection and people who develop chronic viremia after reinfection mount 2 distinct CD8 T cell responses. In resolvers, CD8 T cells are more polyfunctional, less exhausted. There is also a higher frequency of cytotoxic Mait cells and an upregulation of IL-32. While in people with chronic infection, virus-specific CD8 T cells are exhausted. They have more oxidative phosphorylation. We don't see a lot of Mait cells, and we don't see the expression of IL-32 either. For our future directions, we want to assess the longitudinal expansion of Mait cells and changes in their functional profile relative to HCV-specific CD8 T cells. We want to also assess the adjuvant-like effect of Mait cells, and we want to evaluate and investigate the effect of IL-32 on both the effector function and the exhaustion signature of HCV-specific CD8 T cells. So with that, I would like to thank my team and our collaborators, and thank you for your attention.
I think we have time for one question, if you stick around. Or we'll take two questions.
Mine will be quick, I think. Very nice. Did you include the TCR clonotyping in your single-cell RNA sequencing data to see if you could measure the breadth of these HCV-specific T cells?
The breadth?
Yeah, so the diversity of T cell receptors used in your sorted CD8 T cells.
Yes, I mean, we have the TCR data. So the thing is that each sample was stimulated with different HCV peptide pools. So we can see, like, we can know the specificity of those T cells, but we cannot compare between samples to see if resolvers are more, like, broad than chronics. I hope I answered your question.
I mean, I think it sounds like something you're planning to do.
Yeah.
You knew the H of A, and you can start to make some assumptions on the clonality of the PCR response.
Yes.
Hi, Nicol. Thank you for a great presentation. I'm Ayub Mahassin from University of Toronto. I just had a question with regards of your workflow. So for the AIM assay, for how long were you stimulating for, and do you expect that there would be like some phenotypic, some transcriptional changes, I mean, when single-cell RNA sequencing, how would you like control for that?
Yes, so we stimulated for 18 hours. So yeah, there might be changes in the transcriptional profile of the cells. However, we decided to use the AIM assay because it allows us to measure CD8 T cells specific for the whole HCV, for whole HCV peptide pools, instead of just looking at specific antigens in tetramers.
Okay, thank you. Okay, so we'll move to the last talk of our session. I'd like to invite Farnaz Aligolighasemabadi. I'm very sorry if I pronounced your last name wrong. And she'll be presenting on characterizing the resistance barrier of HCV vaccine-induced Antisera. Please.
Okay. Thank you for the introduction. I'm Farnaz from Memorial University. I'm doing my graduate program in Dr. John Law Lab. And today I will be talking about how we characterize the resistant barrier of HCV vaccine-induced Antisera. First of all, I would like to thank the organizing committee for giving me this opportunity to present this work. Okay. No, this is the second one. Okay. So I wanted to start with the fact that despite having effective antiviral treatment, there's still no preventive vaccine for HCV. And one reason is that HCV is extremely diverse, and vaccine need to work broadly. Here we have a virus with E1E2 glycoprotein on its surface. These glycoproteins are essential for viral entry, and also they are target of neutralizing antibodies. Within E1E2, we have different regions that can be targeted by neutralizing antibodies. And researchers found neutralizing antibodies like ACH4, AR3A, and AR5A that can target these regions. So because these regions are conserved, these neutralizing antibodies can target different strains. So despite genetic diversity, E1E2 still contain neutralizing domains that can be targeted across multiple strains. This tells us something important. An effective HCV vaccine should target these regions to provide a broad protection. Based on this rationale, previous studies used the recombinant E1E2 to induce immune responses. In a particular study by Dr. Wang et al., they showed that the recombinant E1E2 from genotype 1A can induce cross-neutralizing protection. And also, as shown here, they showed that the Antisera from immunized goat could block the binding of neutralizing antibodies. So this suggests that E1E2 vaccine-induced Antisera contain antibodies that can target multiple neutralizing epitopes. So we are using this Antisera to define the protection against HCV. But the story doesn't end there. We know that even though the antibody can target epitope, the HCV can find a way to escape. As shown here, there are different mechanisms that HCV escape from neutralizing antibodies. For example, HCV can escape by direct epitope mutation, or HCV can shift the glycan and shield the neutralizing binding site of antibody. So the antibody can no longer bind to that epitope. And also, HCV can escape by local or global conformational changes on its E1E2. So the antibodies are ineffective to bind the epitope. Also, in previous studies, escape mutations have been identified against monoclonal antibodies. As listed here, there are different mutations that can confer resistance to monoclonal antibodies. Like N417S mutation can confer resistance to AP33. So we ask a question. A key question is that if the virus can escape antibodies, can vaccine-induced Antisera still neutralize the viruses with mutations? So to answer this question, we made the viruses with mutations. We identified these mutations as listed here. These mutations confer resistance to different broadly neutralizing antibodies. And then we introduced these mutations into the virus by site-directed mutagenesis. So we have viruses with, we have viruses carrying different mutations, different monoclonal antibodies mutations. So the virus that we use has a luciferase reporter to quantify the infection. Before testing the neutralization, first we confirm the infectivity of these variants that have different mutations using luciferase assay. So the data showed that all these mutants that has mutation retain robust infectivity well above background level. And then we assess, the neutralization of vaccine-induced Antisera or monoclonal antibodies to see if the virus can escape. We used the Antisera from the goats that were immunized with E1E2 vaccine that developed by University of Alberta. First, we tested the monoclonal antibodies. Here we have, on y-axis, we have percent neutralization, and on x-axis, we have the increasing amount of AR3A. So we see that the wild type neutralized by AR3A, and at highest concentration, we have a strong neutralization. And as the antibody diluted, the neutralization decreased. And then we tested the mutant that has mutation against AR3A. As shown here, it showed a rightward shift in neutralization curve, which indicates that more antibody is needed to neutralize this mutant. And also it confirms that this mutant with this mutation, D431G, can confer resistance to AR3A. Also, we tested other monoclonal antibodies like HC84, AR5A, and AP33. Each mutant showed a rightward shift in neutralization curve, which means that these mutants are are resistant. Yeah, are resistant to corresponding monoclonal antibodies. And then we tested our question to see if vaccine-induced antisera can neutralize these mutants. As I said in previous slide, all these mutants were confer resistant to the corresponding monoclonal antibodies. And vaccine-induced antisera neutralize all these mutants and retain the neutralizing activity against all these mutants. So we found that even though the virus has mutation, the vaccine-induced antisera still neutralize it. And then we wanted to know if what happened if the virus accumulate multiple escape mutations. So we made the virus with multiple mutations, and each of these mutations confer resistance to monoclonal antibodies like AP33, AR5A, and HC84. And also we used AR3A as a control because the virus doesn't have any mutation against it. So the antibodies results showed that this mutant that has different mutation confer resistance to the monoclonal antibodies that it had mutation against. And when we tested the AR3A as a control, the mutant was sensitive to AR3A. So this tells us that this mutant with multiple mutation confer resistance to the monoclonal antibody that it had mutation against. And when we tested the vaccine-induced antisera, the data showed that wild type and mutant both neutralized by vaccine-induced antisera, and antisera retain neutralizing activity across this mutant. So we find that even though the virus has multiple mutations, the vaccine-induced antisera can still neutralize the virus. This effect was not limited to just a single animal antisera. Also, we tested the guinea pig antisera from 8 animals. We tested 2 mutants that had multiple mutations, and tested the guinea pig antisera. Each antisera showed strong neutralization, and both mutants were neutralized by a guinea pig antisera. So even though the virus has multiple mutations, the antisera, the E1, E2 antisera can neutralize the virus. To conclude, I would say that our data showed that if the monoclonal antibody can target one epitope, and if that epitope mutate, the virus can escape. But vaccine-induced antisera immunity is different. The antisera contain different antibodies, and these antibodies can target multiple epitopes. So if the epitope mutate one or more, if there is one or more mutation in the epitope, vaccine-induced antisera can still neutralize the virus. So this tells us that vaccine-induced antisera can create a high escape barrier, and also this supports the E1, E2 glycoprotein as a promising HCV vaccine candidate. For the next step, we want to let the virus to select the escape under antisera pressure. So I will do the cellular passage to see if the virus can escape. So if we find any escape mutation, it means that we identify a resistant mutation, and also we can inform the next generation vaccine design. And if we don't find any mutation, it indicates that the antisera create a high escape barrier and durable protection against HCV. So both outcomes will give us valuable insight into E1, E2 vaccine strategies. It brings me to the end of my presentation. I would like to thank my supervisor and my colleagues at Memorial University of Newfoundland, and also Dr. Houghton and his team at the University of Alberta for providing the antisera. And I end with the glimpse of St. John's, the signal hill where the first transatlantic signal was received. And the other one is the colorful jelly beans houses in downtown of St. John's. And we are happy to invite you to visit St. John's. Thank you for your attention.
We have time for one question. Justin?
Great talk, really interesting. I may have missed it, but could you talk a little bit about the background into which you introduced the mutations? Was it a single strain? And do you think if you had done the same experiment in a different strain, maybe you would potentially get a different result?
Mm-hmm. So I introduced this mutation into the J6, JFH1, that this genotype doesn't have any HBR1, and we have a luciferase reporter gene inside this virus. So that's a good point that we can try this for other genotypes to see if we can get the same result. But as long as these antibodies from this Antisera can target the broadly neutralizing epitopes, so I believe that this Antisera would work for other genotypes as well.
Thanks.
Thank you again for the talk.
You're welcome.